Okay, 3, 2, 1, it's jam.
What's up, science fam? The cell is really basic. It's the fundamental unit of life. We can't get enough of it. We always need more cells. In fact, there's lots of research into different types of cells. So, how exactly does the cell divide? How does the body make more cells? What does it take for it to split? My name is Sir Mon, and this is Science with Sir Mon. Let's discuss this. Cell division. How a cell splits. There are two different ways. So this lesson we're going to talk about how mitosis and meiosis are different from each other, and later on we're going to talk about cell transport in another video. Alright? So what does a cell look like coming out of the interphase? It generally looks a bit like this. In late G2, we have duplicated DNA, ready centrosomes, so we're pretty much ready for cellular division. The DNA is copied already and the centrosome is there to split stuff. So let's get right into it. After G2 in the mitotic phase, okay, we have mitosis. It's somatic cell division, meaning it's how body cells will divide. So in mitosis, we have what we call PMAT. It's the four main phases of mitosis. Okay, so it's PMAT. So we start with P, prophase. So in prophase, generally we have mitotic spindle fibers. We have the chromosomes starting to get thick, and the nucleolus is starting to disintegrate so that the chromosomes can be attached to. Okay, we need to free up the chromosomes. In late prophase, we have the breakdown of the nuclear envelope, and we have the fully condensed chromosomes, and the spindle fibers are starting to go into place. Okay, so what are spindle fibers though? How do they work? So spindle fibers are actually fibers created by your centrosomes. So they're created as a form of microtubule complex, and there are three different types. You have the astral microtubules, which anchor the centriole to the edge so that the centrioles can pull or push. There's kinetochore microtubules, which tug at the chromosomes. They connect to the kinetochores at the centromere. And we have the polar microtubules, which push against each other so that there is a interaction between the pushing of polar microtubules and the pulling of the kinetochore microtubules. And that's how your microtubules tend to exert force on your chromosomes. So how are chromosomes arranged by the fibers? How are they exactly pulled to the proper place? So metaphase is the phase where your chromosomes are put into a metaphase plate or an equatorial plate. Now, there is a checkpoint here to make sure that they are in the proper place. It's called the spindle checkpoint. So it checks if the microtubules are attached, the microtubules of your spindle fibers, and if your chromatids are evenly put into the metaphase plate. So if everything is great, if everything goes well, it's time to separate your chromatids. So they are separated in the anaphase. Your polar fibers are made longer to make them push against each other, while your kinetochore fibers are made shorter to make them pull the chromatids apart. So they're actually, the long microtubules are being made longer or shorter. So you're adding more segments or removing segments from them. So since the chromosomes split, now they have to be repacked into the nucleus, and that's the point of the telophase. It's also known as the reverse prophase because the cell is like it's going back to normal. The spindle fibers disappear, the nuclear membrane is starting to come up again, and the chromosomes start to get thin or decondense. So another stage that's not part of mitosis but is necessary for cell division is cytokinesis. So cytokinesis is basically the splitting of the cell. So in animal cells, we have the formation of a cleavage furrow, while in plant cells, we have a cell plate. So since plant cells have a cell wall, they can't just split apart like animal cells do. They just form a new cell wall called a cell plate. So what about sex cells, like your sperm and your egg cell? How do they get made? They can't be made through mitosis, since mitosis has the same number of chromosomes, diploid to diploid. For meiosis, what happens is your somatic cells divide into gametes, or your sex cells. So from diploid to haploid, half the number of chromosomes. So what's different? How does meiosis work? What makes it different from mitosis? There's two parts. We have meiosis 1 and meiosis 2, and each of them has their own PMAT process. In meiosis 1, your PMAT process is different because it introduces genetic variation. So meiosis 1 has special processes that make variation a thing and gives your sperm or egg cells a unique genetic combination. Alright, so each sperm cell and egg cell is unique from others, even if it's from the same parent. So in prophase 1, you have crossing over. Your homologous chromosomes, you have pairs of chromosomes, one from dad and one from mom. They pair up together, okay, and they cross over just like they did when making you. So they get on top of each other and they exchange genetic material, just like mom and dad used to do. So in the crossing over, this is what actually happens. There's a point of crossing over called the chiasma, or in plural, chiasmata. So that's the point where they cross over and exchange the genetic material. So you can see here the exchange of genetic material. The genes are different. Okay? They exchange between the two chromosomes. Now, for that to occur, we need to have a synaptonemal complex. It's how the homologous pairs link together. Instead of actually just crossing over their chromatids, they actually get on top of each other, just like mom and dad. Okay, so protein complexes form as they're on top of each other so that they can switch without any problems. Now, in the next phase, metaphase 1, there is what we call random orientation of your homologous pairs. So what that means is that since your homologous pairs are attached to each other, in metaphase they can be oriented so that the dad side is on this side, or it could be flipped so that it's a different orientation. You can see it in the diagram in the background. You can have this orientation where the red ones are on the right, or you can flip one of them where one of the red ones is on the left. And what happens is that during anaphase, the combinations of chromosomes will be different in the end. So you have just 2 different situations and you have 8 different combinations at the end of meiosis. And since there's only 2 pairs of chromosomes here, imagine if you have 23 pairs of chromosomes. There's so many different combinations that can happen due to the random orientation of homologous pairs. So these 2 different processes create so many unique combinations for your gametes. So anaphase 1 and telophase 1 are pretty much meh. It's just the same kind of process. The result being meiosis 1 produces 2 haploid cells, both with duplicated chromosomes, meaning their chromosomes still have 2 sister chromatids. They still look like an X. You can see here in the diagram, it's still X-looking chromosomes instead of being single chromatids like in mitosis. So what's the point of meiosis 2 then? What does it do? Since your chromosomes are still duplicated, they still have 2 sister chromatids, meiosis 2 just makes them unduplicated. It's just the PMAT process that separates the 2 chromatids from each other. Again, it just splits the chromatids so that it becomes unduplicated chromosomes. So, how many daughter cells in total from meiosis 1? We have 2 daughter cells. In meiosis 2, we have 2 more daughter cells from each one. So 2 times 2 is 4 daughter cells in total. So meiosis produces 4 haploid cells in total. You can see here it's compared with mitosis. Mitosis produces 2 diploid daughter cells, while meiosis produces 4 haploid cells. But is it the same for sperm and ovum or egg cells? Do they have the same process of meiosis? Generally, no. There's lots of sperm cells, but only one ovum at a time, and that's because of your gametogenesis. So spermatogenesis is for your sperm. So each meiotic process produces 4 sperm cells. You can see here in the diagram. So we start with a diploid spermatogonium. It undergoes mitosis to become a spermatocyte, primary spermatocyte. Then it undergoes meiosis 1 into a secondary spermatocyte, and meiosis 2 will produce the spermatids or the sperm cells. So again, take note of these different terms at different stages. First mitosis, then meiosis 1, then meiosis 2, and it changes name between these phases. So it's pretty much normal meiosis for sperm cells, but for your egg cells or oogenesis, we have 1 ovum and 3 polar bodies. So I'm going to get rid of my face. The polar bodies are kind of not so useful, so they just undergo apoptosis at the end. The ovum, however, is the one that gets ovulated as the mature egg cell. So what happens in oogenesis is developmental arrest at prophase 1. So what that means is that in meiosis 1, at the start of meiosis 1, your egg cell does not continue in the meiosis process. Why? That's why girls have menstrual cycles. That's why there's only 1 ovum at a time. So they all get arrested at prophase 1, the immature ova. Okay? So when the menstrual cycle gets to the ovulation phase, just one cell goes into the rest of the meiosis process. So again, at ovulation, girls tend to have just one egg cell that finishes. So it starts at prophase 1 again, and then it finishes all the way until the mature ovum is secreted at the end of, at the end of meiosis II. So again, it only finishes in time for ovulation. All right? So cellular processes need to adjust to fit the processes and needs for the organism. So for guys, since we just kind of yeet our sperm there, we just produce as many sperm cells as we can in as varied ways as we can. While for girls, since they have a menstrual cycle and they need to prepare for pregnancy, they only produce one ovum at a time. So how do you relate to this to us? How willing are you to adjust to the unique demands of these times? Okay, sometimes you need to hustle, sometimes you need to take a rest and wait for the appropriate time, and it's up to you in order to manage your time wisely. All right, so that's pretty much it for this video. I'll see you guys next time.
Party's over.